Indian Academy of Sciences was established by Sir C.V. Raman in 1934 to promote progress and uphold the cause of science in the country. Raman Chair was instituted in 1972 by the Government of India through its Department of Science and Technology in 1972 in memory of Sir C.V. Raman. A highly accomplished and eminent scientist from all over the world is invited by the Council of Indian Academy to occupy the Raman Chair each year, connecting her or him with the Indian science community and society at large. This being the 90th year of the Indian Academy of Sciences, we are delighted to have two Raman Chair positions. One is occupied by Professor Sarah Teichmann, who is with us today, and she comes from the University of Cambridge, and Professor Aviv from MIT. Professor Sarah Teichmann is the Chair and Professor of Stem Cell Medicine at the University of Cambridge. She is a biophysicist and bioinformatician engaged in development of computational methods to explore structural biology, genomics, and single-cell biology data. Professor Regev is a computational biologist and system biologist. Sarah and Aviv are co-founders and co-leaders of the international consortium Human Cell Atlas. To get started with the interaction session now, I invite Professor Teichmann to make a presentation on the history of Human Cell Atlas, an international consortium which will stimulate discussions and interactions with the audience. Over to you.
Thank you, Umesh, for that kind introduction, and thank you everyone for coming this morning. It's an incredible pleasure to be here and an honor to be the Raman Chair for this year with Aviv. And I'm delighted to talk about the Human Cell Atlas first in the next 10-15 minutes or so. So imagine if we had a map of the human body, not organ by organ, not tissue by tissue, but single cell by single cell. So a map of the human body, cell by cell, for 37 trillion cells, approximately, in the body. This map has the potential for scientists to engineer cells for research purposes, to design therapeutics, and to treat disease. But to take advantage of this potential, we first need to understand the molecular basis of the cells in our body, and in fact, we need to define what the cell types are that are inside our tissues. And once we achieve this, we have the potential to restore tissues and engineer cells in the dish, and that would be a revolution. This may seem like an impossible mission. So how do we do it in the Human Cell Atlas consortium? Well, first, a little bit of history. The idea of mapping cells isn't new in the Human Cell Atlas community and has been around for a long time. So for instance, in Sidney Brenner's 2002 Nobel lectures over 20 years ago, he said we need a problem of making maps of cells and maps of how cells talk to each other, so how they communicate. And he called this the Cell Map Project. And he said for this, to do this cell mapping, we don't need a model organism. And what he meant by that is that we can just study ourselves, the human body, because we can. That's kind of the basis for medicine. And he said this will be one of the things to occupy us for the next few decades. And of course, around this time, 2002, we had the first draft of the human genome publicly released. And that DNA sequence of the human genome is exactly the same in every single cell in our body. It encodes for roughly 25,000 genes, and of course, which genes are active in every cell, which subset of those 25,000 genes are active in the cell, is key to defining whether a cell is a muscle cell versus a neuron versus a cardiac cell, and so on. So it's the subset of genes that are active that we really want to understand. And we need to know that subset. And what's helped us is that since 2009, we've had a revolution in genomics which allows us to measure the active genes cell by cell. Single-cell genomics was a resolution revolution because for the first time it allowed us to sequence the active genes in every cell. And so that basically allows us to then determine in a given tissue where we have a heterogeneous subset of cells in the upper versus lower respiratory system, the heart, thymus, brain, etc., in an unbiased way, what are the components of the genes in a tissue. And single-cell genomics is such a revolution because it allows us to resolve in an unbiased way the different cells within a tissue. And of course, the sort of workflow for this technology involves handling the tissue samples, handling the human tissue samples. So here, in a schematic way, what we're illustrating is basically taking cardiac muscle. Then you can either dissociate the individual cells or extract nuclei, then analyze them with whichever single-cell genomics technology you want. And of course, what that yields is a huge amount of data in the sense of the different cells or nuclei and which of the 25,000 genes are expressed at what level in each of those cells or nuclei. And so that data analysis, processing, integration, interpretation is a key task kind of in this endeavor. And it's really the statistical methods, the AI, the machine learning methods that have made this possible to then identify, to cluster and annotate the individual cell types in a tissue sample. So single-cell genomics gives us the parts list of the cells, as it were, the sort of list in a tissue sample. But where are the cells positioned relative to each other within that tissue? Here, it's single-cell genomics combined with spatial genomics methods, spatial transcriptomics, by either imaging or sequencing, that tells us and allows us to define the relative positions of the cells and give us, as Sidney Brenner said, more information about those cell-cell interactions. And so it's the combination of the single-cell genomics and these spatial methods where we take a thin tissue section and then basically get the sort of landscape or geography of gene expression in that thin tissue section that then gives us the sort of atlas, in a sense, of the X and Y coordinate systems of the locations of these constituent cells in the tissue. So these technologies have emerged over the past 15 years or so, which have helped us basically towards this endeavor of atlasing the cells in our body. But remember, I said there's an estimated 37 trillion cells potentially on average in a human body, and so that's a huge amount. So it's more than a single lab or collaboration could achieve. And to have the critical mass to really achieve a mapping of all the cells in the body. In 2016, with Aviv Regev, with Partha, with many others, we founded an international consortium called the Human Cell Atlas. And now I just want to take one minute to pause and to point out that we are really in this spatial biology era and spatial genomics era. So just earlier this year in June, we had a meeting in Stockholm at the Royal Swedish Academy of Sciences about spatial biology in the genomics era. And Aviv and I gave a talk in the last session, as you can see up there, about applications of technologies. And of course, that's really important for the Human Cell Atlas initiative, because now these technologies are so mature that we can really apply them at scale in terms of spatial transcriptomics and multiplex imaging methods. So the mission that we sort of define for the human cell atlas as a community is to create a comprehensive reference map of the types and properties of our cells, because of course the cells are a fundamental unit of life. I mean, just like genes and proteins, they're the constituents of our tissue. And this is a basis for a deeper understanding of our physiology, our tissue architecture, ultimately sort of a new anatomy. And it also has applications that are really becoming a reality now for diagnosing, monitoring, and treating disease. Even at the very inception of the project, Aviv and I and others were absolutely committed to having equity, ethics, global collaboration, and open data access as sort of tenets of the way that we do this project. You can see here a reference basically from Partha and others from the World Health Organization Genomics program about these sort of principles for implementations of human genomics and of course also cell atlas technologies for human health. In terms of these priority areas, what have we done? We formed working groups for both equity and ethics from the very early stages of the project to basically help us track metadata standards, metrics, running capacity building programs, and for the ethics, which is important because of course we're handling human tissue samples, we need to have the correct legal ethics and governance framework, and we provide toolkits for researchers for the patient information sheets and consent and so on. So how many members do we have now across the globe? What you can see here are the institutes that are involved across the world. We have over 3,600 members. So we're a grassroots, scientist-driven initiative. This isn't a top-down initiative determined by funders or anything like that. This is really an open initiative with over 3,500 members. We've now recently counted almost 140 in India and 120 institutes. And so anyone can join. Basically, there's a link at humancellatlas.org that I'll show later. So this international community is also interdisciplinary, and I want to emphasize that, of course, we need the clinician members because for human tissue sample access, we have clinicians and clinician scientists who are members and work with biologists, with the genomics technologists, physicists, engineers, and of course also the computational data scientists, statisticians, mathematicians, in terms of the data analysis and interpretation. So this is not only an international initiative, it's also an interdisciplinary community that's working together. And there are scientists from Asia that are leaders in the community. I've mentioned Partha Majumdar, also Shan Sparkman in Singapore, Sanjruti Saha in Bangladesh is now involved in pediatrics analysis, and many others. So as I've mentioned, this is really open to anyone interested in mapping the human body. And for more information, go to www.humancellatlas.org. So we founded the initiative in 2016, and we're now 8 years on, approximately halfway, let's say, estimated through the project. We've obviously had the pandemic that was unexpected, that hit us sort of in between. But that actually helped us to show how powerful the human cell atlas can be in transforming our understanding of the body, not only in health, but also in infection and disease, and how this can really lay the foundation for a new era of precision medicine, and also guiding the engineering of cells in the tissue. In my lab at the Cambridge Stem Cell Institute, we're making cellular maps of many organs, from the lung to the gut to the heart, and the immune system. And we're also mapping human development in embryonic and fetal stages during pregnancy to understand how our bodies are made at the earliest sort of stages. And we've also mapped what goes wrong in various human diseases, and I'll give a short example here. One of the recent examples is the heart, for instance, which has revealed many unknown aspects of the cells that live in the heart, and also in disease mechanisms like cardiac fibrosis. So just to emphasize again the community aspect of the project, we had an HCA meeting here in India in Kolkata, organized by Prof Junior in November last year, almost a year ago. And there were over 400 members there, as you can see, from Asia, but also other parts of the world. And the benefit of having this global community is that we can share lessons learned, work together to support scientific advances. And as I've said, if you're interested in joining, all it takes really is to sign up to the short questionnaire at www.humancellus.org, join-HCA. And the sort of a short history is that we, as I mentioned, we were founded in 2016. We had a meeting with about 100 members in October 2016 in London, and sort of formed the nucleus of the organizing committee, which basically kind of coordinates the whole project, and then the first working groups. We then have these three broad phases that we can kind of see now. One is data generation and establishment of that community that I've described. So the first phase was really focused on a lot of single cell, suspension cell, and nuclear data generation. And now we are roughly in this broad second phase of assembling all that data and integrating it and making draft integrated data objects available on data.humancellus.org. And we've already had the lung, the brain, the eye, and organoids, and these have come out in these different publications, but also provide integrated reference objects, data objects that we put on the portal. And we're now in parallel also entering a third phase where we're expanding the atlas, making more comprehensive data sets and focusing a lot on spatial biology, which, as I've mentioned, is now really entering kind of the sort of exponential phase. We've mapped about 100 million cells from tissues around the body, and the way we've organized the community of the data kind of integration and annotation is into these different biological networks that we formed. Each one has a couple of coordinators, and then research groups that are interested in those organs and tissues and systems. So we have different organs like the liver, the lungs, the brain, the heart, and so on, and then also systems like the immune system, and we also have a working group for organoids, which is basically taking the in vitro generated systems and comparing them to the in vivo tissues in the body. And just to emphasize again the importance of AI/ML kind of in this project, none of this data would be possible to analyze without machine learning methods. And of course, we're now in the era of foundation models like ChatGPT that are basically models that can fulfill multiple different tasks. So you can ask ChatGPT to answer a question, you can ask it to make a poem, you can ask it to generate a text. And in the same way, the community is generating foundation models for at the moment, mostly based on single-cell suspension data, but there are also multimodal models that link different layers of information, like images from H&E pathology to spatial transcriptomics. And so what we hope in the long term is that the human cell atlas data will be used to train a foundation model for the human body, basically incorporating multiple different layers of information from the single-cell transcriptomics, or the RNA, which genes are active, but also single-cell epigenomics, the spatial data sets, and then the histology imaging that goes with each one. And sort of looking forwards from now, basically into the future, as I've mentioned, we want to work towards a complete version 1 atlas, including the cell annotations, so kind of have spatial maps in these tissue sections, but also assembled into 3D with a z-axis, to work towards creating a globally representative atlas with kind of diverse samples from around the world, so representing human populations, and also have improved use and access, basically in terms of things like foundation models, web portals, and so on, because that's so important for really realizing the benefit of the project kind of across the world and also across communities into the medical community and so on. So I want to finish by emphasizing that the human cell atlas is already enabling major biomedical advances. in rare disease, because the atlas is basically a guidebook in terms of genetic variants, so which cells express genetic variants, and therefore how you expect those variants to penetrate. In terms of tumors, for instance, where there's so much cellular heterogeneity, and basically comparing the tumor of a given tissue to the healthy reference data is very informative in terms of understanding what's the cell of origin, what are the mechanisms of immune response to the niche, and so on. Also, common diseases like IBD, where do the risk variants, where are they expressed in which cells, and again, how are the cells changing relative to the healthy reference tissue, and then also infectious disease such as COVID-19, which I'll come on to in the next part of the talk at 11:30. I want to also mention that there are also human cell atlas members that are addressing questions of interest around the world, oral cancers in India, gallbladder disease in Latin America, and of course Partha, Shyam, and others who are involved in the Asian Diversity Atlas, looking at blood cell composition, basically using single-cell methods from large numbers of samples and populations around the world. And I'll just finish this first short kind of history sort of overview talk right here, and happy to take any questions before the tea break.
Yes, sir.
This noted with math, the number of scientists in Russia seems to be low, not just compared to the population, but compared to the experts in the field. Really profit in getting North Korea and Russia independent.
Maybe we should add, I mean, it's completely open, so everyone is free to join. We're not, blocking anyone, so it's really, up to them to engage.
Some of the Russians have joined in the Asian.
Yeah, I saw 7 lakh, but I thought just looking at the number of scientists available, I could be.
So these are fairly expensive projects. So and we're supposed to generate money from within. So it becomes a little bit restrictive in terms of our ability to participate.
For helping me. It's a team effort.
Yes.
After the street test, I have a very simple question. Early days of the consortia, which you mentioned about, you are talking about a very, very large number of cells. You're talking about the whole diversity of cells. When you take them out from a tissue, their positional context is gone. I understand living in the spatial transcriptomics, but I'm sort of chewing on the first part a bit more. So what generates a lot of data about what kind of transcripts are in each of these cells.
Yeah.
In those days, how easy or difficult it was, by getting to know what one finds, to put them back in the context. So if you pick up a cardiac cell, obviously one would get to know way more diversity than one would get to know just histology.
Yeah.
Teasing the tissue out.
Yeah, it's a great question.
How good is the reverse part, bioinformatically, then and now?
I mean, then, we try to sort of bootstrap our way by integrating the very deep and rich single-cell genomics data with lower plex technologies like standard immunohistochemistry, human protein atlas, low multiplex single molecule FISH, also using clues just from the histology of where cells could be, but it wasn't giving that kind of approach, which I'll mention briefly in the next talk, in the early project that we worked on at the center. Wasn't entirely accurate. Now, then we were in a phase where we had spatial transcriptomics either at kind of 50 micron resolution, which is not single cell, it's more like 20, 100 cells or so. And then the approach that people have used, me and others, is deconvolution, using probabilistic kind of models for integrating those low resolution 50 micron RNA sequencing of the sort of landscape of cells with the suspension cell data. And then what you get is, of course, the localization at that resolution of the 50 micron, not at the single cell. I mean, the other approaches have been sort of higher plex imaging, like with the Murfish or Seekfish and so on, which have been a few hundred genes, and then that has given much better ability to impute, so the single cell resolution but lower plexity, and then impute from the expression patterns of the small number of genes, the whole transcriptome into that spot, into that cell. And so there have been kind of like iterative sort of computational methods that have tried to get higher and higher kind of resolution, accuracy, depth of localizing the cells in the tissues in that way. And of course now we're really getting towards genuine kind of single cell spatial transcriptomics, at full transcriptomic depth with various imaging-based and sequencing-based methods. And that's really the ultimate kind of technology.
Hello, excellent talk. I just had a question regarding sort of links to what Sir asked earlier. How do you ensure, do you capture like demographic data as well from the cell analysis that you're doing? Is there sort of, looking into environmental sort of effects and all that?
Yeah, that's a great question, and I mean, Partha can add some more on that. So it depends a lot on the project how much sort of metadata there is, both kind of what you're saying is demographic, environmental, but also clinical healthcare records and so on. So in some projects there's a lot of depth, in some there's less, and then obviously because of data privacy, the ability to release that also varies depending on projects and geographic location and regulatory environment and so on. And so in the human cell atlas community, what we've been working on is having sort of metadata standards, so that there's kind of like minimal levels of metadata for the released with each data set, or then sort of tiers of then deeper metadata. Yeah. But clearly it's incredibly important because in the long term, particularly in things like where you have these large data sets of many people, you want to be able to correlate the cell profile with the environment exposure and so on.
The mapping process, when you're saying from molecules to the cell, there are various steps, there are a variety of things that you have to look at. How do you decide which organs you have to look? I'm sure it's not complete in all the cases, in most of the cases. Yeah. Probably it comes to some level, matching up to something else.
So we've basically operated in a sort of federated way across the project that everybody works on whatever they are interested in, whatever they want. And so what that's resulted in is, let's say, especially with COVID, there was like a ton of interest in the airways and the lung because it was clear that it was important to understand the lung, and there's a lot of interest in the brain, particularly in the US with the NIH Brain Initiative, a lot of funding. I mean, the funding also plays a role. The other thing that plays a role is accessibility, so blood. I mean, that's what the largest data sets are for blood because it's so accessible. So it's achieving comprehensive coverage is a challenge because of those factors. And we try to encourage by basically also having these biological networks and sort of, tracking coverage, but it is a challenge because we're not imposing that.
Essentially, wherever you're getting data, you're trying to make use of it.
Yeah, exactly. That's how we're operating, yeah.
So also in terms of design, we are actually targeting specific organs. So it's not just we're collecting random data and then putting it into organs. No.
Yeah.
Well, because internal organs are much more difficult to sample. And, from a tissue we can gather some cells and do a large number of cells, but to get tissues from multiple people, so between individual variation is more difficult to assess than within individual variation between cells. So
that's also another factor. But for the internal organs, actually, in the way we get our samples is through deceased transplant donor program, where we have ethical approval for harvesting or for getting biopsies from this single incision from the neck to the waist. But there, that gives us, a cardiac, respiratory, gastrointestinal, and so forth. But for instance, getting towards the spine is much harder because the surgeons don't have enough time to get in. So in the operating theater, they have 20 minutes. After death, the organs are taken for transplantation. And because the theater time is so expensive, they only have 15-20 minutes to give us the research biopsies after the transplant. So there are lots of kind of technical issues. So the spinal cord and the dorsal root ganglia and the autonomic and peripheral nervous system are much less covered right now in Human Cell Atlas, and that's also because the access is really hard. Yeah.
So we have one question for us.
Other than just contributions to the medical field, what are the other significant applications of cell mapping?
I mean, there's obviously basic science and just understanding how the body works. There's medical applications. We are now linking to understanding environmental exposures in terms of, for instance, pollutants, toxins, and so on. So we had a recent meeting on, studying, for instance, organoid systems and profiling by single-cell genomics after addition of different toxins, environmental toxins. So there can be also kind of an interaction with environment, climate, and so on and so forth.
If students have no other question, let me ask one question. So can this data be helpful in understanding the mechanisms of, let's say, metamorphosis or biology, the independent evolution can be visible?
Yeah, so in the next talk, the first part of the talk, there'll be one part of the talk that's on the placenta and first trimester pregnancy, and basically we do study samples from embryonic and fetal stages of pregnancy using tissue from terminations, from social terminations. So the regulatory environment in the UK allows us to do that. And what's super interesting there is also, like you said, kind of computational inference of the developmental relationships between cell states. But actually, we also have those relationships in our body in adult stages because there are stem cells in our blood, in our epithelial tissues, and so we can also compute sort of the developmental trajectories in terms of the similarities between cell states from the adult stem cells to the differentiated cells. So there's a lot of scope for that in Human Cell Atlas.
Sounds very exciting.
Thank you.
If there are no other questions, please join me in thanking Professor Teichmann once again. Came in his slot where we were supposed to go.